The broadcast is now starting. All attendees are in listen-only mode.
All right. Good afternoon, good morning, good evening, everyone. Thank you for joining us today for this very special presentation. This is a bioinformatics analysis with iPathwayGuide, major characteristics and drug repurposing for severe COVID-19 cases. I think we should just go ahead and get started. The presentation today covers work that was done in April 2020, so a little more than a year ago, right at the beginning of the COVID-19 pandemic. And it was described, this work has been described in 4 recent papers with that will be linked at the end of the presentation. What happened is that some next-gen sequencing data was analyzed, some surprising results were found, and then those results were validated in a clinical study. So today what we're going to do is introduce the background for this therapeutic area, demonstrate the analysis that was undertaken in order to find these results, put those results in context, and then show you the results of the clinical study. What we'd like to have you do, you are currently in listen-only mode, but we want to make this an ongoing discussion. Dr. Moore and Dr. Dragic are going to be going back and forth in this discussion, and we want you to join us as well. If you have a question, please enter it in the questions section in your control panel, and I will keep an eye on those throughout and interject them as appropriate. So feel free to participate in real time, but we will leave some time for questions at the end of today's presentation. Okay, so without further ado, I'd like to introduce our speakers for today. Dr. Gil Moore is a world-renowned expert in immunology. He currently holds an endowed chair and is scientific director of the Mott Center for Human Growth and Development. We are very honored to have him join us and share his expertise with us today. Dr. Sorin Dragic is also a world expert. His bioinformatics research lab has created algorithms that have been cited thousands of times and are in use by scientists around the world, including 9 of the top 10 pharmas. Dr. Dragic is Dean of the Anderson Institute at Wayne State University, co-director of the Bioinformatics Core at Karmanos Cancer Institute, and coincidentally is founder and CEO of Advaita Bioinformatics. So without further ado, take it away, Dr. Moore and Dr. Dragic.
Very good. Thank you so much for the introduction and the invitation to participate in this web seminar. I'm sure some of you will ask, what do we need to still talk about COVID-19 and discovering new therapies, understanding the disease, when really we are coming out of this pandemic? Unfortunately, and I really mean unfortunately, although the data is showing that the cases are decreasing, in many places in the world, the cases are just going up. So that indicates that we are not over, because this can expand, and especially because of the risk of mutations. Consequently, it's extremely important that we maintain our awareness of this disease and try to find new therapeutic approaches. So what is COVID-19? I'm sure many of you, or the majority of you, are familiar with this concept. Is the disease that was defined by the infection with SARS-CoV-2, which is a beta coronavirus, and it's very similar to coronavirus who were already infecting humans in the past, such as SARS-CoV-1, the original SARS-CoV, as well as the MERS. The next. Where it comes from, I know that you read in the news and in the media, and there is a lot of potential sources and origins of this virus. The data, the science suggests that 99% of what has been of the sequence that was found in the humans correlates with a virus that infects the bats. So there is the suggestion that somehow this was originated in the bats, and there is a host switching, and there are several hypotheses of what is the intermediary host species. I'll show you in a minute before it reaches the humans. It can be transmitted by person to person, and now we know that. I mean, there is no doubt via airborne and induce what is defined as severe, respiratory syndrome. Next. An important aspect, and we will go through that when Dr. Dagrisi start analyzing the data, the virus has the capacity to inhibit the type 1 interferon response. We can expand that if there is questions and trying to understand what is the meaning of that, but it's critical for the development of the disease. And by inhibiting type 1 interferon responses, enhance the potential of bacterial complications. The next one. So this is an important aspect, and again, there is a lot of things going on in the media. It is a positive sense single-stranded RNA virus, and because it's an RNA virus, it has the capacity to acquire a lot of mutations if it's passed continuously. It is thought to be originated in the bat. Next one. So it is next one, and the intermedial component is not clearly defined. And again, there is a lot of hypotheses. Until now, and now the numbers can be significantly higher, is 52 million cases worldwide. And again, that is in a small number compared to what may have been in reality, because not everybody was tested. And there is 1.3 million deaths. So it's a real pandemic that has affected the life of all over everybody in this globe. What is the entrance of roots? The virus, as I mentioned at the beginning, is transmitted by through the respiratory channels. It goes through the mouth or through the nose, reach the trachea, and will infect the cells in the alveoli. That is the main target, the initiation target, the alveolus. And again, the alveolus are the units in the lungs that are responsible of the transfer of oxygen from the blood into the veins and the arteries into the circulation. If those alveolus are damaged, the individual cannot bring the blood and the oxygen to the body, and that is much of the symptomatology that we will see. So the lungs are infected. There is an immunological response in the lungs that is associated with what we call an inflammatory cascade with the production of a lot of cytokines. And the cytokines, really what they do is, by trying to protect other cells to be infected with the virus, start killing the cells, start inducing the cell death of the cells. Unfortunately, when you kill the cells in the alveolus, you are killing the units that has the capacity to obtain oxygen and it transfer to the red cells, leading to what the patients will develop, that is problems with obtaining air, a respiratory syndrome. However, although much of the data that you may have heard and the main symptoms that the patients are diagnosed is the respiratory component, there is a lot of evidence suggesting that is damage or complications in the gastrointestinal tract, so nausea, vomiting, diarrhea, the brain, headache, impaired consciousness, confusion, and there is also a cardiovascular problem, especially in the long-term effect. A lot of patients who had COVID-19 are describing complications after the disease, especially associated with the brain and the cardiovascular system. Next one. What is the classical clinical evidence that a patient had the infection? Is looking at MRI or CT scans in the lungs. The presence of what is called, that you can see in the figures, in the upper figures A and B, those patches, those white patches that exist in the lungs, yeah, those are the classical signature of the disease. Next. So let me just summarize quickly these two aspects. The lungs belong to what we call the mucosal immune system, because they are continuously exposed to viruses and bacteria. When we talk about a mucosal immune system, we are referring to a layer of epithelial cells who have the capacity to identify, recognize pathogens such as virus and bacteria. If a cell is infected with a virus, like in the case of COVID-19, the cell will start producing cytokines, will induce a response in order to stop the replication of the virus. The cytokine storm that has been described in the cases of COVID-19 is a strong mucosal response, recruiting immune cells from the peripheral blood into the site of the infection in order to stop viral replication and also to stop the dissemination of the virus in the body of the patient. That is the primary objective of the mucosal immune system. Recognize, identify, respond, and recruit immune cells to prevent the dissemination of the disease. The next one. How it recognizes? It will recognize through the expression of a specific receptors in the mucosa that is called the toll-like receptors. And again, I'm not going to go through the details, just to give you the message. The toll-like receptors, when it recognizes the virus, it will activate a cascade that leads to the production of pro-inflammatory cytokines. I repeat again, those pro-inflammatory cytokines will activate immune cells, will recruit additional immune cells, and will promote potentially the death of infected cells. Next one, and I think that is my last slide. So that is a general understanding of what, in a very short period, of what is the virus and what is doing in the cells when infected. So having this general knowledge, we wanted to understand more details. And the objectives of the study that we're going to share with you is the following. We wanted to have a better knowledge and better understanding how the epithelial cells, this mucosal immunity, is able to respond to the infection by SARS-CoV-2. And how this response is related to many of the secondary complications that we are seeing in patients infected in COVID-19, such as the brain, the liver, the lung is the primary, the cardiovascular, and so on that I mentioned to you. And identify putative mechanisms involved in the disease. And the last one, and I think is the critical aspect, we need to identify better therapeutic approaches when the patients are infected. Let me just finish with 2 words. We do have vaccine. Yeah? The vaccine is very good and it protects. But unfortunately, we still have a lot of patients who either are not vaccinated or will be exposed to mutations. The vaccines in those cases, of course, are not working. Consequently, we need to improve the therapeutic approaches. And that is the tools that we are going to be discussing today, how we can prevent or treat those patients infected with SARS-CoV-2. Dr. Dagrisi, I'll pass it to you.
Thank you. So just to clarify the format, so we're going to switch back and forth. So Dr. Moore gave you this brief introduction. I'm going to show you some data sets that we analyzed. We're going to jump into some analysis. We're going to find some things there. We're going to go back to Dr. Moore and let him explain and elucidate what is happening. So we're going to switch between the software and the slides a couple of times, so I don't want you to get confused. So we started with a couple of data sets. So again, the time frame here is end of March last year when the first RNA-seq data sets became available. So we had some data from primary lung epithelium infected with SARS-CoV-2 versus MOC. And we also had A549 cells. This is a cell line that were infected with a couple of different things. One was, of course, SARS-CoV-2, but also RSV as well as IAV. So this is to try to understand a little bit what is specific to SARS-CoV-2, the response to SARS-CoV-2 versus what would be a general response to respiratory viruses. We also had data from COVID-19 patients. So we had uninfected human lung biopsies versus COVID-19 patients who actually died of the disease matched for age. So those were the first data sets that we started working with. I'm going to jump into the software here. Let me just make this bigger. So the software we are using here is called iPathwayGuide, and is designed for exactly this kind of analysis. So you have several phenotypes, and you try to understand them. We have some folders here. I'm going to go into the COVID-19, and I'm going to open the data set that I just mentioned. This is actually a meta-analysis involving all those data sets that I mentioned, but I'm going to just start with one of them, which is, so those are the phenotypes that I described. Lung epithelium infected with SARS-2 versus control. This is A549 infected with IIV, CoV-2, and so on. I'm going to start, I'm not going to walk you through everything, just highlights. I'm going to start with the COVID-19 versus healthy because it's kind of the most relevant and interesting. But the point is, I wanted to give you the background that we looked at various comparisons and tried to understand what is specific to this particular infection versus other similar infections, and also across various type of tissues, including patient tissue and lung epithelium, whole lung, as well as cell lines. So here on the top, you have various tabs, and they go from simple to more complex. So genes, GOTAMs, we're going to look at biological processes, then pathways, upstream regulators, and so on. I'm going to start with genes. So the classical approach is in such a high throughput experiment, we have two phenotypes, is let's look at the differentially expressed genes, the most differentially expressed genes. So we can do that in this graph by, this is the classical volcano plot. You have log fold change here, positive and negative. So this is upregulated in red, downregulated in blue, and this is minus log of fold changes. So the ones to the left and right and to the top are the most significant. So I mouse over, I see the gene, I click it, and unfortunately, there is not much known about this particular gene. There is a description, but it's not involved in any known pathways, no drugs are targeting, and so on and so forth. So these are all the annotations. It is known that it has these molecular functions, but no specific biological processes. So we don't learn much here. If we go maybe the other down-regulated genes, the most down-regulated gene is this RAD. There are some biological processes, molecular functions, but no pathway information, no drugs. And there are some SNPs and microRNAs. But the point is that just by looking at the genes, we don't really understand much. Yes, there are genes that are differentially expressed. Yes, some of them are differentially expressed by 8-fold, 10-fold. This is in log scale, so this is quite a bit. But just by looking at individual genes, we don't really understand what's going on. So we have to become a little bit more sophisticated. Let's look at biological processes. When we do this, there are various ways of doing this analysis. The classical way is kind of enrichment analysis. You look at the number of differentially expressed genes that are associated with a given process and compare that with what you expect by chance. We're going to correct for multiple comparisons. And if we look at the top, we're looking at the top 10. Let's look at the top 25. We see a lot of things that make sense. Immune response, immune system process, defense response, innate immune response, and so on. So all these things are very they give us a very reassuring feeling, because that's what you expect to see, response to virus, response to stimulus, and so on. So, but it all makes sense, but it doesn't shed any light. And unfortunately, this is often the case with this type of analysis. And if you look at it, there are tens and tens of such processes. The list goes down, and over 100 biological processes that are still significant. So this is the classical approach. So we need to, again, do something a little bit different. So let's look at this in a different way. This is a diagram that shows the relationship between these biological processes, their similarity based on the number of differentially expressed genes that they share. So for instance, the first 22 biological processes identified share 66 differentially expressed genes. And we can zoom in here, and let's look at this a little bit more closely. This is the top biological processes: immune system response, immune response. It's 10 to the minus 24, 10 to the minus 21, and so on. But let's look around. So neutrophil mediated immunity, neutrophil activation involved in immune response, neutrophil activation, neutrophil degranulation. So clearly we have, the data tells us that neutrophils play a crucial role. So I'm going to just ask you to remember this, and let's do one more thing. Let's do a different type of analysis, which looks for very, very specific biological processes. And now we are going to see, I can go back to the counts, or I can zoom out, but let's look at this: negative regulation of viral genome replication. So this time—
Before we go to that, may I ask you, so the difference between the two approaches that you just used, yeah, in terms of the zooming in and out, in your opinion, what is more effective?
Great question. So the first one is the classical approach, which is just based on the number of differentially expressed genes. And if you look at the dendrogram I showed you, you see that many processes share many things in common, many differentially expressed genes in common, which is very redundant. And that's why you have so many biological processes that are significant. So as I mentioned here, for instance, 22 GO terms have 66 differentially expressed genes in common. The second one, the high specificity, it's a proprietary approach. And if you look here, these are it. Number one, you have much fewer biological processes, and now you have very little redundancy. So here you have only 11, and if you go the equivalent thing, you have 0 genes in common. So this looks down in the gene ontology hierarchy. You see it goes about 7 or 8 levels down, and it gives you very, very specific biological processes that are likely to be much more informative than the one that you obtain with the other type of analysis. Yeah, this is a proprietary method that is available here. Did I answer your question?
Perfect, thank you.
Okay, so the most interesting thing here is the type 1 interferon signaling pathway. So this is, again, is very, very specific. It's very deep down in the GO hierarchy, and it's one of the things that I think, as Dr. Moore mentioned earlier, is going to be crucial in this thing. Again, we still see things that are defense response to virus, innate immune response. Everything makes sense. Again, neutrophil degranulation, neutrophil chemotaxis. So, and if we want, we can look for if there are any other neutrophil-related things, now that we have a pointer. And we see that there are not fewer than 6 biological processes that are related to neutrophils. So this being said, I'm going to switch back to the slides. And these are the first 2 things that we learned from the data. First, oh, sorry. Let me go back and one more step. Let's go to pathways. So far we learned about neutrophils, and we know that the type 1 interferon is important. Let's look at the pathways. Here, the top most impacted pathway is cytokine-cytokine receptor interaction. And this is, again, a little bit different from the way many people do it. Many people just look at the enrichment on the pathway as well, but this is very limited because essentially the enrichment approach uses just the number of differentially expressed genes and ignores the position and role of the genes on the pathway, the signals, and so on. We use a different approach, and we add this perturbation type of evidence. So we have enrichment, but we also have perturbation. So we want the pathway to be here because that means there is a lot of evidence of enrichment type as well as a lot of evidence of perturbation type. Let's look at the first pathway. This is cytokine-cytokine receptor interaction. So again, the role of cytokines in infection, it was known, and Dr. Moore just briefly mentioned it. But let's see what happens in this particular COVID-19. We see there are a lot of chemokines, a lot of cytokines, and they are all up. So all these interferon alpha is right here. So cytokines, chemokines, everything that is pro-inflammatory is severely upregulated. If you look at these genes here, these are the genes on this pathway, and all but a few of them are upregulated. And if we look at the ones that are downregulated, let me if you look at the ones that are downregulated, you see that they are exactly the ones that are supposed to control the immune response. So essentially, the ones that are pro-inflammatory are upregulated, and the ones that are anti-inflammatory, that are supposed to regulate the inflammation, are down. So this is essentially a freight train going down the hill with the gas pedal, the floor, and no brakes. So this is a clear image of hyperinflammation and essentially a cytokine storm. If we look on the second one, okay, I'll go back to the slides for a second so that we can talk a little bit about this. So we learned a couple of things here. We learned about the fact that neutrophils play a crucial role, and there is a hyperinflammation cytokine storm. So Dr. Moore.
Yes, so this is fascinating because we know that the patients have a lot of cytokines, but we don't know exactly what are all those cytokines. And in general, in immunology and in every biology, we know that there is a balance between the inducers and the inhibitors. So what you found, Serene, is fascinating because it's just showing us that when you remove the brakes, then the cells are massively responding to the presence of the virus. There is another aspect that is fascinating here, is again proving that the mucosal immunity, because you are analyzing the epithelial cells, so that the mucosal cells, these epithelial cells who are target, they are also able to respond, and they are the ones who are bringing all those immune cells, and they are producing the cytokines to recruit all these immune cells. And the graphic that I have here is exactly to make that point, which correlates to what you just showed, Sorin. The virus infects the epithelial cells, but what I said before, they are not just sitting there or doing their classical role of getting oxygen. They also are a mucosal immune cells. And when they are infected, as you are showing us, they are secreting a lot of those inflammatory cytokines who are going potentially, we know, they will go to the blood. And when they go to the blood, they will do several things. One is they will activate immune cells. Second, they will start calling them towards the tissue that is infected. Or they will start also sending to other tissues where potentially the infection is taking place, including outside of the lungs. Now, a cytokine storm, unfortunately, it doesn't limit to the organ where it's infected. The cytokine storm is going to affect the whole body, depending also on the magnitude of that cytokine storm. So the activation of the cytokines are taking place first in the lungs, but, and there's a big but, it will start disseminating into other organs through the circulation. And that cytokine storm may be activating other type of immune cells in different organs, which may lead then to what we call the multi-organ failure or a hyperinflammation syndrome. If we are not able to control that hyperinflammatory syndrome, the outcome is the death of the patient. So let me remind you, the majority of the patients who died of COVID-19, they are dying because of this systemic inflammation that is damaging multiple organs. The next one, sorry. And there is an important aspect that you mentioned and you show, the neutrophils. Yeah? Why the neutrophils? And that is the beauty. When you show us the data, I was looking also to understand more about the neutrophils. This is incredible cells because they have a very short lifespan. Yeah? And they are one of the major anti-infection cells. So what do they do? And very quickly, when a cell is infected, they are like airplanes with bombs. So they will identify those infected cells through the toll-like receptors or through the cytokines produced by the epithelial cells that are telling them, I am infected. They will go to the infected cell and they will start releasing a specific substance that will try to kill the infected cell. So that was the classical mechanism that we knew about the neutrophils. However, and some of the pathways that you show us suggest that there is a little more than the classical role of the neutrophils. These neutrophils, the factors or the pathways that we saw that are activated there are suggesting that the neutrophils are undergoing cell death. So why would you want that the main defense or the cells associated with the main defense will die? For a simple reason, and not so simple. When the neutrophils die by a process of netosis, they will release DNA, and I think I have another picture in the next slide. And this DNA that is released start forming what is called the NETs, neutrophil extracellular traps. And what is the role? It's a fascinating process because the main role, or they commit suicide in order to create these nets, and the nets are like the fishing nets. They will start stopping all the viruses or the bacteria that has escaped the original infected cell, and consequently will prevent that the virus will infect another tissues or the bacteria will infect another tissues. Yeah. So what we are finding from this analysis is that neutrophils are involved in the response to COVID-19 or to SARS-CoV-2, and moreover, they are dying, and they are dying for formation of these nets. And indeed, there are now several reports showing that one clinical characteristic of patients with COVID-19 is neutropenia, that it means a decrease in the number of neutrophils. And the next one.
Dr. Moore, before you keep going, we have a question.
Yes.
Which is, what pattern recognition receptors play important roles besides toll-like receptors here?
Thank you for the question. So let me just go quickly. The toll-like receptors are in the surface or intracellular. In the surface for bacteria, intracellular for viruses. There is another type of receptors like the NOD receptors, as well as a family of intracellular receptors that will recognize either double-stranded RNA, single-stranded RNA, which are the type of genetic information coming from the viruses. There is also receptors who will recognize in the family of the nodes, who will recognize double-stranded RNA, which is originated from DNA viruses. So again, because of time, I cannot go to more details. Just to give you the concept, you have surface receptors, toll-like receptors, and you have a family of intracellular receptors who their function is to recognize different components from the virus, from viral infections, either DNA or RNA viruses. I hope I answered your question.
Thank you.
So let me finish just with this concept. The formation of these traps of from the neutrophils, we know now is an important, a critical mechanism for stopping the dissemination of the infection. However, and there is a however in everything, although this mechanism is defined in order to stop the virus or the bacteria to continue infecting other cells, it also may induce a process of coagulation or clotting. Yeah? And if we go to the next one, that's where I will finish my summary of this, because the major problem that we see with patients with COVID-19 is that they start developing coagulation problems. And what it means, coagulation is associated with the activation of the platelets, activation of neutrophils, formation of the nets that I told you, activation of the cascade that is associated with the clotting system, and closing of those blood vessels. Yeah. If the closing of a major blood vessel happens, so let's go to the next one, it leads to many complications associated with the clotting system. Yeah. But at least from the analysis of the data that Dr. Dagreschi was showing us, clearly suggests that with the longest infected with SARS-CoV-2, there is an inflammatory response associated with a cytokine storm, is activating neutrophils. The neutrophils are forming these extracellular traps, and the extracellular traps, by stopping the virus, protects that the other organs will infect it, but unfortunately may lead to clotting problems. Go ahead, I'll pass it to you back.
Okay, so let's jump back into the software and see what else we can see here. So hopefully it didn't time out, but just to refresh. So let's look at the second pathway here. And now we can take advantage of this capability that we have to calculate the perturbation at every point in the pathway rather than just a statistic for the whole pathway. And if we do that, we can show you coherent cascades. A coherent cascade is essentially a putative mechanism. It's a sequence of steps in which the expected changes according to the pathway match the observed changes. So let's look here. So we see this very nice mechanism that starts with cytokines, and look how many there are that are severely upregulated, and then the receptor itself is upregulated, and then it goes to JAK and STAT. And on the same pathway, we have this other mechanism which appears to be isolated. So it involves this gene here, PTK2B or PYK2, but it's kind of disconnected. So we wonder what happens. According to this particular pathway, which contains everything that is known, they are not related, so they are not connected. So what we can do, we can construct a network from scratch, ab initio, and see if we can see something else in this one. I'm going to hide the isolated nodes, and let's look for that those genes. So they appear to be here. This is STAT, JAK, and this is PTK2B. So in reality, there is a known interaction. So JAK3 actually activates PTK2B. Let me just make this bigger. Hopefully you'll see it a little bit better. So this edge here, which is kind of not present on this pathway, is the one that makes the connection. So let's see what kind of evidence is there for this. I'm going to click on this edge that tells me that is the JAK3 activates PTK2, and here we have the evidence for it. This is coming from a curated data, so the confidence score is extremely high, is the maximum. And here is the paper, and I can actually have a quick look at the abstract, and yes, it says exactly that JAK3 activates that PTK. So I'm comfortable with this. I believe this is true, and that makes sense because now essentially you have one single mechanism on this pathway, and that is this chemokine receptor JAK-STAT. Let's look at upstream regulators. Well, we can since we are here, let's look at influenza A, for instance. Influenza. This is the pathway that describes the influenza, and it has a similar mechanism. So here is the interferon alpha, which is, these are all interferons, but the alpha is upregulated in our COVID-19. And again, through the JAK-STAT IRF9. So this is something that happens in other viral infection, not only in COVID-19. We can look, for instance, very quickly at the toll-like.
The influenza is also a RNA virus, so it makes sense what you're finding.
Right. Thank you for that. So those are the other pathways that, and you see they are all significant that Dr. Moore mentioned, toll-like receptor and nod-like receptor signaling pathways, and also other virus infection. But let's just go to the upstream regulators to see if there is any gene that could explain this, that would act as an upstream regulator. And it finds, correct for multiple comparison, and there are essentially a couple of genes that, but there are two that are extremely highly significant. Why? One is IRF9. I'm going to show you what happens in a different layout. And now you see why this is identified as an upstream regulator. So essentially, IRF9 regulates several genes, 37 in total, out of which 17 are differentially expressed in our condition. And if you look at it, so this IRF9 is known to activate this particular gene, and indeed the gene is up, and so on. So this is true for all but one. IRF9 is known to downregulate this gene, and indeed this gene is downregulated. So that all is evidence that supports the hypothesis that IRF1 is active as a regulator in this condition. Let's look at the second one, STAT2. Let's use the same layout, and we see that this is essentially taking the phenomenon one step upstream, right? So yes, IRF9 is regulated, but also STAT2 has a regulatory effect, activating all these genes, as well as IRF9. And again, we can look at the evidence for this particular interaction, and we see, there are quite a few papers, and if we look at the type of experiment, there are various different types of experiments that point to the same thing. So this is a 2-hybrid experiment. This is affinity capture, another affinity capture MS, reconstituted complex, and so on. So there are quite a few papers supporting that. So this seems to be real again, so I can trust this. So this is something that is very interesting, and is particularly interesting because you would have missed this with the classical analysis, because this STAT2 doesn't meet our criteria. Yes, it has a fold change of plus 2.2, but the p-value is not significant. So that's why initially it was not included on that graph with differentially expressed genes, and you see how important it is, right? And 17 out of its 18 immediately downstream genes are changing in a manner that is consistent with the hypothesis that this is upregulated. So this is very interesting. So we identified two upstream regulators, IRF9 and STAT2. Let's see if we can do something about drugs. Now we're going to look at drugs and their effect on these differentially expressed genes. I'm going to look for drugs that are predicted as absent. What does that mean? That means that if I were to consider this drug and administer this drug to the patient, this drug would have the ability to revert the gene expression changes that are measured. Okay, so I'm going to look at those, and I'm going to look at in a different layout. Well, maybe it's too much, but let's look at some of the drugs that are on top here. So you see what happens. This is well, let's look at the top one. Tofacitinib is in the same layout. So you see what happens. So this particular drug would act directly upon all these genes. These genes are differentially expressed, and in particular, they are upregulated in COVID-19, and tofacitinib would downregulate them. This sign here shows you this is inhibition. So this drug would downregulate all the red genes that are upregulated in this condition, and would increase the expression of this particular gene, which is downregulated in this condition. So it would revert the changes. So this is kind of very interesting. So this is a JAK inhibitor. Let's look at the second one, diclofenac. This is a NSAID, so a non-steroidal anti-inflammatory agent, and it does something very similar. You see, every gene that is downregulated by the disease would be upregulated by this drug. So those are the blue genes, and they are all would be reverted. And all the genes that are upregulated in the disease, they will be downregulated by the drug. So that's the kind of thing that we want to see. Why did I not take the first one? Well, the first one acts only about on 18 out of the 19 genes, whereas this one gets and the next one, methylprednisolone, has much more targets that would be reverted by this. So, but let's just look. I'm going to just show the drugs only, so not other chemicals. And let's just look at the top 3 or 4 drugs here. So tofacitinib, diclofenac, methylprednisolone, gold sodium thiomalate. So this is another anti-inflammatory used in RA. Then this is another steroid. Okay, so those are the top 5 drugs. So I want to jump into the meta-analysis, which brings into the picture the other cell types and data that we had. I'm going to look at the drugs directly to see which of these drugs would be consistently seen in the same type of analysis in other tissues. And I'm going to just sort by p-values and show the drugs only. And I'm going to look for drugs that would revert the changes. A little bit is changing. I hope you can follow this. So I'm looking at the drugs that are predicted as absent. So those would be drugs that would revert the gene expression changes measured in the disease. And these are this is COVID-19 whole lung. This is lung epithelium infected with COV2. This is influenza, and so on. So there are various types of this, and you see methylprednisolone is top, and the top 5 are the same: gold, sodium thiomalate, prednisolone, tofacitinib, diclofenac. So this is remarkably consistent. They are all significant in all this. Going back to the slides. So what we have done here, we have looked a little bit at mechanisms. We have identified upstream regulators, and we were able to identify some drugs, specific drugs that would be effective. We also identified the type 1 interferon plays a key role. JAK-STAT was the mechanism, and the 2 upstream regulators were STAT2, IRF9, and the drug that we chose is methylprednisolone. Why? Because it's widely available. It is, and it will be widely available. It's cheap, and it's very effective. It reverts a large number of genes. So we then proceed to go on other data sets, completely independent from the ones that we did this analysis. So now we have, again, various types of data from cell lines, tissue culture, as well as patient samples. And this time we have Vero86 cells infected with SARS-CoV versus control, lung organoids infected with SARS-2, intestine organoids. So these are all now publicly available in GEO. So I don't want to jump into the software, but I show you in this new data set, completely different tissue, completely different labs, everything is completely different. You see the same drugs. The top 5 are exactly the same. You can always sort them, but gold sodium thiomalate, anti-inflammatory, methylprednisolone, and so on. This gold sodium thiomalate is an anti-inflammatory used in RA, but it's not really widely used in the US. So methylprednisolone continues to be our top drug. So we reached this conclusion in kind of April 2020. So as I said, we chose methylprednisolone as being a drug that we wanted to just focus on. There was a small problem though. The small problem was actually a big problem, and that was World Health Organization. This is the clinical guidance as of 2020, May 2020, and the World Health Organization, with all their scientists, recommended against the use of corticosteroids. So we had a problem. What do you do when you have a problem? Nobody believes you. Well, you do a clinical trial. So this was undertaken at the Henry Ford Health System, led by Dr. Mayur Ramesh. This is a multi-center health system in Southeast Michigan. There were 213 patients diagnosed with COVID-19. 38% received the conventional therapy, 62% received the methylprednisolone, and we looked at getting worse. So if a person is admitted in hospital and has to be moved to ICU, or if it is in ICU and the treatment was given and then it has to move on mechanical ventilation, or if it is on a ventilator and then it dies, right? So all that was a composite endpoint, and the results were very significant. We also look at mortality. So the mortality was amazing, was from 29.6 to 16.6%, so a reduction of about 44%. And this is the 30-day all-cause mortality. So if they went home, got out of the hospital and they were hit by a bus, that still counts here. But that's a classical way to look at the data. So this is 30-day all-cause mortality, and there's a huge difference between prednisolone and methylprednisolone and the standard. Soon after, there was a trial coming from the UK. People looked at dexamethasone, and the results are very positive. That's another steroid that worked, but there is a difference. The number needed to treat for dexamethasone is 8 for patients on ventilators, but 25 for patients needing oxygen. Whereas for methylprednisolone in our trial, the number needed to treat is 8. The lower the number, the more effective the drug is. So we still believe that methylprednisolone is more effective than dexamethasone, even though dexamethasone got a lot more publicity. So indeed, the WHO changed their guidance. So now there is a whole booklet, and now they do recommend the steroids for this. These are the 4 papers that present this result. The first one is the data analysis that we've done to identify the drug. The second one is the clinical trial results, focusing on providing all the information about it. This is about thrombosis and vascular injury in COVID-19, and this is again platelets and renal failure. If anybody's interested, we can share this data analysis and let you play. Maybe you can see some other new things. I'll stop here and we'll take any questions. Thank you.
All right, thank you so much to both of you. This was really a wonderful presentation. I'll leave some time for people to submit their questions into the questions box. We do have one that's been waiting. What kind of death do you expect, if not apoptosis, for the neutrophils during COVID-19 infection? Oh, Dr. Moore, I think you're muted.
Yes. Okay, I was muted. Sorry for that. The type of death that is now characterized in the neutrophils is called netosis. And it is a very specific, because it's a type of suicide of the cells where there is going through the classical process of cell death. But the major difference is that there is no degradation of the histones of the DNA. The DNA is a structure to form this this mash. If you go to the next slide, Serene, we can show them. You see, they form. I don't know if you can see you form this mash. In the upper part, yeah, in the upper part is the neutrophil that is dying, and this mash, the green one, is the DNA from the cell with the histones and so on that is forming the mash, and that is what they will be tracking or stopping the bacteria and the viruses, preventing that they continue traveling in the blood. So the cell death is a type exactly like the majority of the cells who died is suicide, but this is very specific because it doesn't break down the DNA. It just fuse them together in order to form these traps, or also now called nets.
Okay, thanks. Can you, I think I followed, but for all of us, can the two of you draw a straight line from methylprednisolone to neutrophils for us? How is that steroid known to act on neutrophils specifically?
Yeah, so the steroids, one of the major actions of the steroids is by decreasing the production of inflammatory cytokines. This is the main role. When you decrease the expression of the inflammatory cytokines, you are then inhibiting the activation of the immune cells. So by preventing the activation of neutrophils, you will prevent also the formation of the nets, and consequently will prevent the formation of the clotings and all what we discussed briefly before. So a central mechanism of the corticosteroids, or the steroids, sorry, is by decreasing the cytokine storm. That is the major aspect, inhibition of the cytokine storm. And I don't know, Serene, if you have a figure there with your analysis.
Yeah, I can. So that's exactly the kind of question that you can ask and get answers for in this tab here. So here you can select your biological process. For instance, let's say neutrophil activation involved in immune response, or neutrophil chemotaxis. Okay, or maybe the intersection between those two. Let's say, or those three. Let's say these are genes that are involved in all these three biological processes. I can build a network with that based on all the known interactions, and I can add methylprednisolone here. So I can say, what is the connection through which, what is the mechanism through which methylprednisolone would impact, if at all, these particular genes. And I've never done this before, so I don't know what's going to come, what's going to do, right? I want to have a little bit more genes though, so I'm going to remove this.
Yeah, because many of the samples. Perfect.
And the answer is there is no direct connection from methylprednisolone to this particular gene. So I'm going to hide the isolated nodes, and you see that methylprednisolone disappears. But essentially, this is methylprednisolone, and the answer is it doesn't act directly on any of these particular genes.
Exactly.
Right.
But if you look at the inflammatory process, you will see potentially that there is an effect in the inflammation, because that's what I mentioned, is an indirect effect. It doesn't act on the neutrophils, but it will act on the inflammatory process.
Great.
Yeah, so I can go, for instance, type in interferon pathway, and I can see what is, and now you're going to see that this is going to be impacting a bunch of these genes. I can move this to make it more clear, but essentially this would be the mechanism. Whoops.
Okay, so can I ask? Do we have time for one more question as we get to the end of the hour here? So one of the here's the question. I noticed that chloroquine bisphosphate was in the top 10 predicted upstream chemicals or drugs. Is that meaningful, with respect to hydroxychloroquine? Remember that whole thing?
Yeah, so actually, we look specifically, and in the paper we discussed a number of other drugs that were considered at that time, including hydroxychloroquine. And the answer is no. The hydroxychloroquine is not expected to do much.
Great. Okay.
On this particular differential expressed genes.
Okay, great. Okay, and I do have one last one. If we didn't get to your question, we will be sure to follow up. But this is a great question. I want to make sure we ask it. Since we may not know all the existing immunological pathways that exist in the body, and the software analyzes only those known and then gives an outcome, how do we account for unknowns?
How do we what?
Account for unknowns.
Oh, so well, that's exactly the point that I was trying to show in the second pathway that I showed. Even though the pathway was incomplete, they did not contain that particular interaction, by constructing a network. So in this chemokine signaling, right? So this particular pathway is no pathway is going to be all the time up to date to the very latest knowledge. But that's how you can construct this this network from scratch based on everything that is known. And this is going to not be limited to the existing pathways or the topology of the existing pathway. And you saw here, this particular interaction was missing on the pathway, but is here. So we can, this is how you go around that problem. You are not limited in any way to the existing pathway or their current topology.
Okay, thank you. All right, we are over time, so it's time to stop. Thank you all for your time today. And thank you both for such a wonderful presentation.
Thank you.
Thank you so much.